So today we're going to talk about something called oogenesis. And it is spelled just like it is said, oogenesis. Okay? What is oogenesis? So oogenesis is formation of mature ovum. Plural word is ova. So ovum or ova if we're talking about more than one. Mature ovum from oogonium. Here's that oo again. And oogonium is the singular. And then if we're talking about more than one oogonium, then we would call it oogonia. And this happens only in females. So in males, you have the spermatogenesis, where you get sperm. And in females, you have the formation of ovum from the oogonium. This happens in two different parts. It happens before birth and after birth. So that's called prenatal development, or during prenatal development, and after birth or postnatal development. So oogenesis starts in prenatal life. So when you are still about to become something in your mother's womb, this starts in prenatal life before you're born. When you are still in your mother's womb, your ovaries start the oogenesis process. So once it is determined that you are a female, as soon as the sperm fertilizes the egg, as you are developing in your mother's womb, this process starts. Okay? As you were developing, some of your most primitive cells, which are going to be germ cells, we call them primordial germ cells. Primordial germ cells. Primordial germ cells appear in something called the yolk sac. The yolk sac is a structure that develops in the beginning of your development in your mother's womb. That's where you get nutrients, oxygen through the blood from your mom, and it also has cells that travel into the developing embryo to become certain cells later on in life. And one of those cells is the cells that are going to be in your ovaries. These primordial germ cells appear in the yolk sac around 4th week of development in your mother's uterus. So your mother is about 4 weeks pregnant when this is happening. She probably doesn't even know she's pregnant yet. In the wall of that yolk sac, the primordial germ cells are found, and they keep on proliferating by mitosis. Okay, we're going to talk about mitosis a little bit later. But right now we want to focus on meiosis. This has to do with the sex cells development. Meiosis has not yet started at this point. These primordial germ cells keep on proliferating and migrating from the wall of the yolk sac toward your gonads or ovaries as you are developing. Again, you're 4 weeks inside your mother's womb. You still kind of look like a lizard. So now we will move our focus and enlarge into what your ovaries actually look like. And we're going to kind of look at a picture that if we cut your ovaries, sliced it in half, what you would be seeing in that development of these cells. So there will be many of these primordial germ cells, of course, but we're just going to take one primordial germ cell and focus on it and then see how that one develops. But there will be many of those developing alongside each other. So let's go back a little bit and talk about a couple of things we learned in a previous lecture. How many chromosomes are present in that cell, and how much DNA does it hold? So we had talked about capital N and small n designation, And capital N, we said, is the amount of DNA. Okay. And then the small n refers to the number of chromosomes present in a cell. So in this particular cell, in your cells at that point, have 46 chromosomes. You remember that? Mm-hmm. Okay. 46 chromosomes. So that is the number of chromosomes. And then the amount of DNA, big N, is 2N, because you get one big N from your mom, one big N from your dad, and that gives us 2N, 2 capital N designation. And then also 2 small n, again, 1 small n from mom, 1 small n for dad. All right? And these are single structured chromosomes. These are just chromosomes that are present in that cell. They haven't replicated themselves, so they're just single structured. So you remember this stuff a little bit?
Yeah.
Okay. So just to repeat, so in these, you have a full set of 46 chromosomes, which are single structured chromosomes, 23 paternal, 1 small n, 1 big N, and 23 maternal, 1 small n, 1 big N. And this is where you get the 2n and the 2n. So this is a regular cell, not a sex cell. And from here, it's on its way to go through the process of becoming a sex cell, which is oogenesis in females. Okay? With a full set of 46 chromosomes, this primordial germ cell has 2 small n designation for the number of chromosomes and 2 big N for the amount of DNA, and it is diploid or haploid, if you remember.
Diploid?
Diploid is correct. So remember, the DI usually means 2, and you have basically the 2 pieces that make up a full complete cell. So that's, you could remember it that way, that's diploid. All these chromosomes are single structured chromosomes still. When these primordial germ cells are multiplying by mitotic division, they are producing the next generation of cells, which are called oogonia. Oogonia. These oogonia, because they are produced as a result of mitotic division still of the primordial germ cells, the number of chromosomes and the amount of DNA is the same as the primordial germ cells that we just talked about. So right now they're just dividing and dividing and dividing and dividing to give you a bunch of cells that eventually are going to go on to become your sex cells. So we're still at the point where mitosis is taking place and just increasing the number of these cells. Okay, and again, the number of chromosomes, the N designation, and the diploid or haploid all still is the same, 2N, 2N diploid. So now, usually, oogonia, those cells that keep dividing and dividing and increasing in number, they will be surrounded by a flat sheet of cells called epithelial cells. So let's say this is your cell right here. So you will have cells, sheets that look like this. And they're connected to each other. So they kind of form like a flat sheet around this cell. These oogonia cells themselves will keep going through mitosis process and go on producing more and more oogonia. So all of these cells now will continue to have the same genetic material. I'm kind of repeating myself a little bit, so just to make sure that right now we're still going through mitosis with these cells. Now, once they have significantly increased their number, a time comes when one of the oogonia starts enlarging and other oogonia around it degenerate. Okay, so randomly one of those will start just to get bigger and bigger, and there is no space for the other ones, so they kind of fade away. And you'll have only one oogonium in a sheet or bag of epithelium cells surrounding it, derived from the surface epithelium of the ovary. Okay, so inside your ovary, some cells will kind of form this sheet around the oogonium. These epithelial cells that are surrounding this single oogonium are called follicular cells. So right here, these are called follicular cells. This oogonium now, okay, that is inside of this sheet of follicular cells, continues to enlarge itself and converts into what we call a primary oocyte. So we went from a primordial germ cell that was dividing and dividing and dividing and dividing by mitosis to an enlarged oogonium. And now, as it continues to enlarge, it converts into a primary oocyte. Once this conversion happens, we now have the cell that will begin the process of meiosis. Okay? Questions so far?
Not really.
Okay, so pretty simple so far. You're in your 4th week of developing inside of your mother's womb. Certain cells from the yolk sac, they travel to the ovaries, and they are the cells that are going to become your sex cells. They are called primordial germ cells. They keep increasing. The genetic material in them is still the same, just like the ones that they came from, 46 chromosomes, 2N diploid. And this sheet of epithelium cells or epithelial cells surround the oogonium, and they're called follicular cells. And now that oogonium converts into a primary oocyte. Now the primary oocyte will start its first meiotic division. Before this meiotic division happens, during prophase 1, DNA will replicate itself. So, as you know, when a cell is about to divide, whether mitosis or meiosis, first is going to replicate its DNA. It's going to double its DNA, right? So now we still have 46 chromosomes, but they are double structured. So that's the difference between going from the oogonium and now into the primary oocyte. It's going to go into prophase 1 of meiosis, and it will have double structured chromosomes. You still have the 23 from mom, 23 from dad, but they are now replicated.
Mm-hmm.
Questions so far? So at primary oocyte, prophase 1, DNA is replicated. So now what is the amount of DNA? Amount of DNA is the big N.
Mm-hmm.
So before it was 2N, and we've replicated or duplicated.
So 4N?
So it'd be 4N, 4 big N. And for the small n, which is number of chromosomes, what would be the number of chromosomes? Or what would be the n, small n?
If it's duplicated?
It's duplicated, but remember, we are still working with the same number of chromosomes. So we never go from 46 chromosomes to 92 chromosomes, because that wouldn't be a human cell. So it's still 46 chromosomes, but the amount of DNA is doubled because we replicated the DNA because we're about to split.
Mm-hmm.
Okay? So the number of chromosomes, does it change or stays the same, or does it get reduced at this point?
When did it say the same?
It stays the same, correct. So it's still 2 small n, and is it diploid or haploid?
Haploid. Or diploid.
It's still diploid. So we haven't split the cells into, 23 and 23, so it still stays diploid. So the only thing that happened is the amount of DNA, which is big N, has doubled, and that's where we get the 4N designation. So here we are during the early stage of prophase in the first meiotic division, after the DNA has been replicated and the oogonium enters into a very important stage called the diplotene stage. Okay? When the primary oocyte reaches the diplotene stage during prophase of the first meiotic division, it arrests its further progression in meiosis 1. It stops. So as a female, when you were in your mother's womb at about 4 weeks of development, your ovaries, or in your ovaries, the cells which are going to become your egg, or eggs later on called ovum or ova, they started their first steps toward becoming those cells. They are not those cells yet, but they started the process. And after you have primary oocytes, and they started DNA, and they finished DNA replication, and your DNA has been replicated, you still have 46 chromosomes, but the amount of DNA has doubled. The cells arrest at that stage. They stop. Okay? And this is called the diplotene stage. All right? So before you were born as a female, all your primary oocytes have started their first meiotic division, and at birth you have no more oogonia. So let me explain that a little bit better. We took an example of just one cell, right? An oogonium that became a primary oocyte and started the process, or started the steps toward becoming your sex cells. And then they stopped at that first prophase in the diplotene stage. So that one is just an example. All of your future to become eggs go through the same process at that point. And at birth, you have no more oogonia that will turn or convert into primary oocytes. Once you're born, that's it. There is no new primordial germ cells that are going to keep replicating or dividing and becoming or converting into an oogonium and then going into primary oocyte to give you more chances at your body producing more eggs. Okay? All of those that will be are done during your prenatal development period. Okay, so at birth, you only have primary oocytes that are arrested in the diplotene stage during prophase 1 of the first meiotic division. Before birth, all the oogonia you had either enlarged and were converted into primary oocytes, or they degenerated. And before birth, all your primary oocytes have entered into prophase 1 of the first meiotic division, and were arrested from further progression in the diplotene stage, and never completed their first meiotic division before birth. So those cells haven't gone on to divide. Okay? Now, here is some interesting facts. About 2 million of these are present in your ovaries when you are born? 2 million primary oocytes. Okay? You actually had about 7 million oogonia, which converted into primary oocytes before birth, right? When your mother was about 5 months pregnant with you, you only had 2 million of them enlarge and convert to primary oocytes. So the other 5 million degenerate. We can say that you were born with all the possible primary oocytes that you will have for your life after birth. There is no further formation of primary oocytes after birth. Remember that all the primary oocytes are surrounded by these flat epithelial cells called follicular cells. Remember that? Mm-hmm. And now we can call it primordial follicle. Now remember what we started with when you were 4 weeks inside your mother's womb. We started with primordial what? Germ cells. Now, after birth, and you have these converted oogonia that converted into primary oocytes, and they started their first meiotic division but arrested at diplotin stage, and they are surrounded by these follicular cells. These are your primordial follicle. Okay, so try not to confuse the two primordial germ cells. These are the early stem cells from where the oogonia will develop, and the primordial follicle, which is the primary oocyte, surrounded by the sheet of epithelial cells called follicular cells. So now we can say that you were born with about 2 million primordial follicles in your ovaries. Now we're going to move on to after birth, or postnatal development. So after you were born, and all the way until you reached puberty, your primordial follicles with primary oocytes remain arrested where they were in the diplotin stage of prophase 1 of the first meiotic division. Following me so far? Mm-hmm. But not all 2 million make it. Okay, so you were born with 2 million out of the 7 million oogonia, right? Actually, most of the 2 million you were born with, they degenerate as well. And around puberty, you probably have about 40,000 primordial follicles with primary oocytes remaining in both of your ovaries. So now you're down to 40,000. Okay? At puberty, when the ovarian cycle or the menstrual cycle starts, a hormone called follicle stimulating hormone. You see that? Follicle. We have a primordial follicle now. So you have a follicle stimulating hormone, FSH. That hormone will come and will stimulate the primordial follicle. Every month in the ovaries, a pool of growing primordial follicles is maintained. Every monthly cycle, about 5 or 15 of those 40,000 primordial follicles start growing. And the follicular cells, remember that sheet of follicular cells around the primary oocyte? Those follicular cells, which are around the primary oocyte, become cuboidal. They become cuboidal, meaning they get a little bit fatter. They start getting fatter like that. So they look like cubes now rather than flattened cells. Now we call this follicle, instead of a primordial follicle, now we're going to call it a growing follicle. Once we start seeing that these flat epithelial follicular cells are becoming enlarged a bit and they look like cubes, they're cuboidal. Now this is a growing follicle. And then they become multi-layered. So instead of them just being one layer, then you will have layers. And when they are multi-layered, we call that stratified. Stratified cuboidal epithelial cells. And at this stage, it is called primary ovarian follicle or primary follicle. All right, so we went from what? We went from primordial follicle to a growing follicle to a primary ovarian follicle or just primary follicle. With me so far? Okay, so now the primary follicle still has primary oocyte or ovum arrested in diploidine stage, but now with a multi-layered or stratified cuboidal epithelium or follicular cells, and it is now called primary follicle. And around these stratified cuboidal follicular cells, a connective tissue starts to differentiate from the ovarian stroma or tissue. During the development of this primary follicle, the primary oocyte itself secretes some glycoproteins outside, and the follicular cells also secrete the same glycoprotein inward. Circle inside, that's your primary oocyte. It's going to secrete outward some glycoproteins, and then the cuboidal cells, the follicular cells around the primary oocyte, will secrete the same glycoprotein. And then these glycoproteins are going to form another circle around the primary oocyte, in between the primary oocyte and the follicular cells. You see it there? And that is called zona pellucida. Zona pellucida is a layer made of a meshwork of glycoproteins surrounding the ovum of the primary oocyte, but inside the follicular cells. Now, with further development of the primary follicle during the monthly cycle, the follicular cells will continue to proliferate, and the follicle will become enlarged, and fluid-filled spaces will appear between the follicular cells, and then they will fuse or coalesce together and form a larger fluid-filled space called antrum. All right, so let me explain. We have the primary oocyte here. We have the zona pellucida around it. And then we have all the follicular cells outside of that. And then we said there was going to be another connective tissue way outside of all of this. Okay? So the cells on the inside will continue to grow, but then there will be some spaces in between them. And then these spaces will come together and form like sort of a space. And that space is fluid-filled. You see all this space is fluid-filled. And it is called antrum. So remember how it looked before? It looked like a ball of cells. And it had the primary oocyte all the way in the middle with the zona pellucida around it. And now the cells have kind of enlarged and left this space inside between them, still encased inside, but it is growing in size, and now fluid is filling this area called antrum. Some follicular cells will remain around the ovum and the zona pellucida. These are called cumulus oophorus. Also now you'll see more prominently the connective tissue around the ovarian follicle. Remember that connective tissue that was derived from the ovary stroma? So now that will become prominent, and we call that theca folliculi. So we have cool names for these things. We have theca folliculi, and these follicular cells that stayed around the primary oocyte Those are called cumulus oophorus. At this stage of the growing follicle, once it develops this cavity, the fluid-filled space called antrum, and the theca folliculi surrounding the structure, it is now called secondary follicle. So now we went from primary follicle to secondary follicle. Remember, this secondary follicle still has the primary oocyte or ovum inside of it, which is still arrested in the diplotene stage of prophase 1 of the first meiotic division. Around this time, out of the 5 to 15 primordial follicles that you started with, only one is left, and the rest degenerate. The theca folliculi layer has two sides. It has an external layer and an internal layer. The internal layer is called theca interna, and the external layer is called theca externa. Later on, we will learn that the theca interna cells, along with the follicular cells that are close to it, produce estrogen. The secondary follicle keeps on growing, and the primary oocyte will come out of its arrested state. Remember, it was arrested when?
At 4 weeks.
At 4 weeks when?
Pregnant.
Yeah, when your mom was 4 weeks pregnant with you.
Mm-hmm.
So it arrested then, and now this ovum is now developing at one of your monthly cycles. So this ovum could be once you hit puberty, or it could be when you're 25 or when you're 30. On any given monthly cycle, randomly 5 or 15 come out and they start growing during your monthly cycle, and only one becomes the secondary follicle, which will eventually become the egg that could be fertilized. So the primary oocyte will come out of its arrested state, and it will complete its first meiotic division. The first meiotic division gives 2 cells. So division, we're dividing them into 2. One that has most of the cytoplasm, called secondary oocyte, and another with very little cytoplasm, called first polar body. So even when that one that gets chosen to be the one that grows and the one that gets taken care of until it becomes big enough to become the secondary follicle, once it divides into one big one and one very little one that doesn't get much of the cytoplasm, but it gets all the genetic material. So now let's talk about the genetic material of the secondary oocyte. So now that we split the genetic material between the one that got most of the cytoplasm and the one that got the first polar body, what is the genetic material state with regard to the big N, the small n, diploid or haploid, how many chromosomes, all of that. So when that first split happens, let's talk about number of chromosomes. How many chromosomes are in each?
23.
23 is in each. So once you see 23, okay, is this haploid or diploid?
Haploid.
Haploid. Very good. Now, what do they look like if you remember? So remember, we arrested and they look like this. And there was 46 of these. So when we split them 23 and 23, they look like they were double-stranded. Right? They were double-stranded DNA that just got replicated and then they arrested. So when they split, even though they are 23 on this one and 23 on this one, are they still double-stranded or are they single-stranded?
Single-stranded, right?
They're still double-stranded. So there is 46 of these double-stranded replicated DNA in the cell when it's a primary oocyte. That's where it arrests. And then when they are separated, when they are divided, you get 23 in each, but you still have double-stranded chromosomes that did not split yet. So that's the first division. We haven't split the ones that are connected at the centromere, right? Those haven't split yet. So they are double-stranded, but the amount of DNA is what? The big N. So we were 4N because we had all 46 and we had them double-stranded arrested. But now we split them. So with regard to the big N, the amount of DNA, what is the designation now? It went from 4N to
2N.
2N. So even though it has 23 chromosomes and it is haploid, because it's double-stranded, the amount of DNA is 2N. Does that make sense?
Yeah.
And what about the small n designation, the number of chromosomes? We said how many chromosomes there are?
23.
And does that give us how many n's of the small n?
Oh, 1N. One small.
1n, very good. So again, even though it is 2 big N, it is 1N designation at this point. It is haploid, and we have 23 chromosomes double-stranded at this stage. So at this point, we give the follicle a new name. So now this is a fully mature secondary follicle. It is called Graafian follicle. So now, second meiotic division starts immediately after secondary oocyte is formed. This is happening around the middle of the cycle, 14 days before the next menstrual cycle. Graafian follicle is fully ready for ovulation. A surge of a hormone called luteinizing hormone occurs to stimulate ovulation. So remember, we had a follicle stimulating hormone, FSH, to begin the cycle, to stimulate the follicle to grow. Now we have something called luteinizing hormone that stimulates ovulation. Okay? Concentration of luteinizing hormone in your blood circulation goes up. But just 3 hours before ovulation, second meiotic division arrests at metaphase 2. Now this ovum, along with some surrounding cumulus oophorus cells, ovulate out of the ovary, and they're taken up by the fallopian tube. Ovulation is your ovary giving out your ovum, your egg. When it comes out of your ovary, it's intercepted by your fallopian tube. Your fallopian tubes are connected to your uterus. All right, so back in the ovary remains what's left over from the follicle. Some granulosa cells and theca cells, which convert into a yellow-colored mass called corpus luteum. Corpus luteum will produce lots of a hormone called progesterone during the second half of your monthly cycle. We'll discuss the hormones in another lecture. The cumulus oophorus cells begin to rearrange themselves around the ovum in a manner that gives the shape of a crown. So now these cumulus oophorus cells, they start looking like this. Okay, so it looks like a crown. This arrangement of the cumulus oophorus cells is called corona radiata. radiating crown, I guess.
Yeah.
Corona radiata. The ovum is still arrested at metaphase 2 of the second meiotic division. It will wait in the fallopian tube from 24 to 48 hours for sperm to arrive. So that's the window. If sperm doesn't arrive, it will just die out and degenerate, and any organic material will be absorbed by the fallopian tube epithelium. If sperm does arrive, and as soon as the sperm touches the cell membrane of the secondary ovum, suddenly the secondary ovum is reactivated. It was arrested in metaphase 2. It wasn't going to divide further, second meiotic division, until and if sperm touches the membrane of the ovum. It is now reactivated, and very rapidly it will complete its second meiotic division and produce one more polar body and becomes definitive fertilized ovum. The first polar body also may divide and become two polar bodies. So you end up with the one definitive fertilized ovum and three polar bodies, which degenerate. The definitive fertilized ovum will have 23 chromosomes. Remember, we went from, now you're going in, during the second meiotic division, you had 23 chromosomes, but they were double-stranded, arrested. Now, once the egg is fertilized and it goes through that rapid secondary meiotic division or second meiotic division, you still have 23 chromosomes, but they're now single-stranded, right?
Yeah.
Okay, so the definitive fertilized ovum will have 23 chromosomes, but they are now single-structured. So the designation for the small n, or the number of chromosomes, is how many? 1 or 2?
Small N or big N?
The small n, number of chromosomes.
1.
1n, right? And now the big N, which is the amount of DNA, what is it now? What was it before, when it was double structured, 23 chromosomes?
2.
It was 2n. So now we have single structured chromosomes. So how many capital N would it be?
1.
1 capital N as well. So that's the amount of DNA. And is it diploid or haploid? Haploid. Very good. Okay, so now the 23 chromosomes from the female form what is called a pronucleus. And the sperm brings the other 23 chromosomes from the dad. Those 23 chromosomes from the dad enter the ovum and form the male pronucleus. So the genetic material that is carried by the sperm is released into the ovum. So the ovum had its own 23 chromosomes, and it formed the female pronucleus, and then the sperm brought the 23 chromosomes from the male, and that formed the male pronucleus. All of this is inside this one egg or one ovum. So both pronuclei will fuse together and become one nucleus. And when they become one nucleus, now how many chromosomes do we have inside of the new nucleus?
46.
46. So the 23 from dad and the 23 from mom now are combined. Okay. And once this happens, how many small n's? That's the number of chromosomes. It was 1n. Now it's 2n because you brought the ones from the father. So it's 2n. And then for the capital N, which is the amount of DNA, It was 1N, now it is 2N. Is it haploid or diploid? Diploid. And now this is called a zygote. And the egg has been fertilized. At fertilization, 3 things happen. So as soon as the sperm fertilizes the egg, second meiotic division is completed, like we just said, and another polar body is thrown out. If sperm brought an X chromosome with it, it will be female. If it brought a Y chromosome, it will be male. So sex is determined at fertilization. Genetic sex. Sperm brings some molecules that are not yet known, but they are known to activate the metabolic machinery of the ovum, and in turn, the zygote becomes fully metabolically active as well. If it successfully develops into a human being, because of the arrival of sperm, this ovum may live 70, 80, 90 years, 100. Without sperm, it never completes its first meiotic division and lives for about 24 to 48 hours.